Okay. Hi, I'm a PhD student at Pest and Environmental Adaptation Research Group at University of Melbourne, and my project is to check the effects of Rickettsia viridis and aphid parasitoid interactions. Aphids are considered as an important agricultural pest due to the direct damage by feeding on the plants and also transmitting the viruses, which cost millions of dollars to agricultural industry per year throughout the world. And two main viruses that are transmitted by aphids are turnip yellow virus, transmitted by Mycos persicke, and Bailey yellow dwarf virus, which is transmitted by Rhopodopodus paddy or oat aphid. So in Pergi, we have three aphid agricultural pest species, oat aphid and Russian wheat aphid and Mycos persicke or green peach aphid. So what are the endosymbionts that we are working on? There are two different types of endosymbionts in the aphids. One of them is obligate or primary endosymbionts like Bachnera that are required for the aphid to survive because they provide essential nutrients for these aphids, and they can only transmit from mother to offsprings, so maternal transmission. So, and the second one is facultative endosymbionts or secondary endosymbionts that they are not really essential for the survival of the aphids, and they can transmit both maternally and horizontally via the parasitoids or host plants. And they have diverse effects on the aphids because they can just protect the aphids against the parasitoids or pathogens or insecticides, and also they can cause the aphid to adapt to different heat or cold situation. And there are over 9 types of the facultative endosymbionts found in the aphids, such as Rickettsia viridis, Hamiltonella defensa, or Regiela insectivora, and etc. So what are the benefits of endosymbionts to agriculture? They can block transmission of plant viruses by insect vector. They can suppress the pest population through deleterious effects such as reducing their fertility or survival, or increasing the susceptibility to insecticides or parasitoids, or change the host plant's suitability. And also they can do some seasonal vulnerability. One of the techniques to transfer endosymbiont across the aphid species is using the microinjection. In the Pergi, we have found that P. aphid or Acetosiphon pisum harbors Rickettsia viridis. So we use this as a donor species and then inject the hemolymph of the P. aphid to the 3 target species of the GPA, Russian wheat aphid, and oat aphid that we got. And then when we got to the stable line, we started to do the parasitism experiment. To choose the parasitoids was, for the green peach aphid, we chose the TLR APA because it is a specialist parasitoid for brassica-feeding aphids. And for the oat aphid and Russian wheat aphid, we chose Aphidius colemani. So when it comes to facultative endosymbionts effects on parasitoids, the most famous facultative endosymbionts in this term is Hamiltonella defensa because it has a huge protection against the parasitoids. And mechanism behind is APSE bacteriophage or Acetosiphon pisum secondary endosymbiont is like Lambda T virus that inside the genome of Hamiltonella, which produces toxin and kills the larvae or egg of the parasitoids, so prevent them from the further development. And also the Hamiltonella affects the aphid alarm pheromone, EBF, and it reduces the alarm pheromone. Alarm pheromone is alarming thing, so it causes the aphid to move away from the danger. And on the other hand, it is like a pheromone, so it affects the parasitoid and it attracts them. To check the parasitism rate of these two parasitoids that we've got, We used, for the first experiment, we used the nymphs of different ages for all three species of aphid that we had. And you can see that there was a significant effects of Rickettsia, and it had a protective effects against the parasites. And for the second experiment, we chose the nymphs of 3 days old to see if there's a difference between, for the parasitism rate and the emergence rate between the infected lines of the GPA and Russian beta aphid to the Rickettsia compared to the uninfected line. And we saw that there is no effects of the Rickettsia this time, and there is no protection regarding the parasitism or emergence rate. And also there was no effects of Rickettsia on the duration of mature stage of the both Rapier or Colemanni. So we hypothesized that maybe the reason for such difference result that we got is Rickettsia density. So we compared the Rickettsia density for both GPA nymphs that we use, 3 days old compared to the different age of the nymphs that we use for both experiments. And as you can see, there is difference between the Rickettsia density. And also to get a clear view of the Rickettsia density and changes across the different age of the nymphs for the GPA, we compared the different ages of the GPA for the Rickettsia density, and you can see that there is gradual increase in the Rickettsia density. When it comes to 7 days old, it really increases a lot. And we also checked for the selection and if there is any preference for selection for both Rapier or Colemanni, and for the between the GPA infected lines compared to uninfected lines to Rickettsia. So to do this, we did some replicates of one female wasp with 5 GPAR plus and 5 GPAR minus, and we recorded them for 35 minutes. And after analyzing the data, we saw that for the Rapier, there is no significant effect of Rickettsia on the number of stabs or the timing of stabs. And also the result is consistent for the Colemanni as well, and Rickettsia didn't affect any of this behavior for the both parasitoid wasps. We wanted to check if the Rapier can transmit Rickettsia horizontally in GPA lines. So we chose, we put one female of the Rapier with a GPAR plus, and then after we washed them for 3 stabs, we transferred the wasp into the new petri dishes with GPA uninfected lines, and then we left the wasp for 24 hours. Then we removed the wasp, and we let the aphids to become mummy. The alive aphids were screened with qPCR for the Rickettsia density, and as you can see, there was a horizontal transmission of 12.5% with Rapier. Also, we checked for the Colemanni if she can just transmit Rickettsia in GPA, and we saw that there is a horizontal transmission of 13.3% in GPA with Colemanni. So to conclude, we can say that protective effects of Rickettsia viridis against parasitism success of paraconid is dependent to Rickettsia density, which varies by GPA curves. And also Rapier and Colemanni can transmit Rickettsia in GPA. There is no preference for selecting green pitch aphid infected or uninfected to Rickettsia in both Rapier and Colemanni. And the use of endosymbionts for biological control of aphid pests will likely affect the interaction with parasitoids. As the next step, we would like to compare parasitism status of Rapier on GPA infected and uninfected one at 2 different life stages of 3 days old versus 7 days old. Once the results are finalized, we will go for the Colemanni to check the parasitic status of Colemanni on Russian beta aphid and Ota aphid. And also we are interested to find the reason for such protective effects of Rickettsia against parasitoids. Also, we wanted to test if there is any maternal transmission of Rickettsia to the next generation. And we would like to establish a stable line of green pitch aphid infected to Regular insectivula via Aphelinus abdominalis. using potato aphid as donor, and also establish a stable line of green pitch aphid infected to Hamiltonella diffensa with Aphidius sanchi using black currant salty cell aphid as donor. And at the end, I would like to thank my amazing supervisors, Ari Hoffman and Parin and Chang, and also other researchers in Perji, Shinwei, Sam, Shufen, Josh, Paul, Katie, and Nick. And this work was a collaboration with between Perji and GRDC and Caesar as ICPIP. And thank you all for listening.
And thank you, Safieh. A fascinating story you've told there, and it certainly sounds as if you've got a lot of interesting work ahead of you with all those next steps. It's very ambitious. All right, let's open things up for a few questions. And you might just have to unmute and start speaking if you've got a question, because I can't squeeze all these name panels onto my screen and see you all if you're raising a hand. Just while we're waiting, Safieh, I mean, these insect microbe interactions are really fascinating, but there's also an applied angle to it, you know, a way in which we can use these interactions to our benefit in pest management. So could you just kind of step us through that, what the steps would be, not necessarily for you and your project, but the steps ahead of us to kind of commercialize or exploit these interactions?
To commercialize? Okay.
How can we make use of these microbes for better pest management?
So if we can just, because we can do the using the micro injection, we can just have the stable line, so we can just sell them to the agriculture. But the point is that if they have the protection against the parasites, so they are not really useful for agriculture. But if they can block the virus transmission, because the virus transmission is more important when it comes to the aphid and agriculture. So if they can block the virus transmission, that would be useful. And I don't know, because it needs more studies to just can go, because maybe in the field, depending on the temperature, it depends and it changes, or we cannot establish the line in the field. So it really needs, and they just transmit horizontally. The wasp will adapt to this after a while. So it really needs more study to can say that we can send them to the field.
Yes, yes, it's going to be quite a long pipeline, so to speak, isn't it? Good. All right. Any further questions before we move on to our next presenter?
Sorry for jumping in. Can I ask a question? A quick one.
Please.
Safieh, very great talk. Just maybe I've missed it. Are those endosymbionts that are being transferred either through microinjections or by the parasitoids inherited into the next generation of the aphids? Did you see any of those effects?
For the microinjection, yes, there is maternal transmission and we have the stable line. But the point is, for the parasitoids, we are checking if there is maternal transmission. And we just saw that there is horizontal transmission. So we are checking if there is, afterwards, we can go from mother to offspring and check how many generations it will stabilize in the population.
Yeah, perfect. Thank you.
Thank you.
Okay, well, thanks, Safieh. We'll move on to our next presenter